All right, so my name is Jonathan Little. This is the Module 3 assignment for cell biology. It's the 25th of April in 2023. And this is on prokaryote-eukaryote interactions. So for this assignment, I decided to talk about archaea because I knew that bacteria interact with us, but I really didn't know about archaea. I kind of just figured they lived down by like thermal vents in the ocean and stuff. But as it turns out, they also make up about 1.2% of our gut microbiome. And so archaea in their structure are very similar to bacteria. They have a cell wall and a cell membrane and cilia and flagella and delocalized DNA in the nucleoid and ribosomes, and they lack, just like bacteria, they lack endomembrane organelles. The difference between them and bacteria is their cell membrane, the fatty chains, the fat chains, the lipid chains have side chains on them, which don't exist in bacterial cell or eukaryote cell walls. And the lipid is attached to the phosphate using an ether instead of an ester. And so archaea, for their size, they're about 1 micrometer, so just around the same size as a bacteria. And then the eukaryote for this interaction is going to be an intestinal epithelial cell. So this is intestinal epithelial cell. We've got some villi here that increase the surface area in the gut. And then we have a nucleus with the DNA. We've got some chromosomes in there. We have a endoplasmic reticulum, the rough part first with all the ribosomes on it. And then we've got the smooth one and that. Well, actually, they're connected, I think. Smooth one. And so rough for the production of proteins and smooth for the production of lipids. We have free ribosomes that make proteins as well. We've got some lysosomes and peroxisomes to help clear out waste and detoxify. We've got a mitochondria in here for cellular respiration to happen. And we've got a Golgi complex or Golgi complex. I'm not quite sure how to say it. It's just hanging out over there. And yeah, so that's the structure. We've got a phospholipid bilayer cell wall. And these are like 10 to 100 times bigger. This is somewhere between like 10 and 100 micrometers. I couldn't find an exact length, but this is somewhere in the range. And so in mitochondria, cellular respiration happens, and it takes oxygen and glucose and turns it into ATP, and CO2 is a byproduct. So a waste product of that. And then additionally, we'll talk quickly about another prokaryote. This is a bacteria, and they do a lot of fermentation in the gut. And fermentation, a byproduct of that, is hydrogen gas. And so these two processes, cellular respiration or aerobic respiration, anaerobic respiration, or fermentation, are critical to the proper functioning of our bodies. And however, if these levels of CO2 and hydrogen gas increase too much, it shifts the equilibrium for the reaction, and the reaction can slow down or even stop. So we have to get rid of the carbon dioxide and hydrogen gas. And so archaea help us do that. Archaea are methanogens, which means they make methane. But really the important part is that they use the CO2 and the hydrogen. And so they take the CO2 and the hydrogen in various concentrations, and they make nope, not CH3, methane, there we go, and water and energy. And so they can decrease the concentrations of CO2 and hydrogen gas, and that allows the processes of cellular respiration and fermentation to continue happening, for proper function. And so, archaea do in fact play a role in our health, and it seems like they haven't been studied all too well. So it's going to be interesting seeing if that may become more important in the future.